The next block is dedicated to the investigator's perspective, and will focus on practical challenges associated with academic ATMP development. It will showcase decentralized manufacturing models, prioritization strategies for rare disease therapies. There will be 3 speakers, and the discussion will be again joined for all of them. And now, the opening speaker of this section is Professor Jürgen Kuball, Chair of the Department of Hematology at the University Medical Center, Utrecht. His presentation is titled Challenges and Hurdles of Academic ATMP Development from Lead to the Market. Professor Kuball is internationally recognized for his work in tumor immunology and engineered immune cell therapies. Professor Kuball, thank you very much for being prepared, and we are ready for your contribution to the conference. The mic is yours.
So I hope I get to watch how long I can speak, because otherwise I will speak for hours. These are my conflict of interest, which are also relevant, because I would like to give you a little bit a different perspective on clinical trials, starting really from an investigator perspective. So some of you I know more from my role in EBMT and making platforms for CAR-Ts and working with companies. But my other life is that I'm writing a lot of patents. We started a company. We may bring new needs to the clinic. And this will I mainly talk about today, because the question is, how do we really bring this into the clinic? And the model we have, just to give you some biology, who's immunologist here? I see no hand. But to make it very simple, we have different immune cells in the blood which control cancer. And one of the blood cells which has not been really appreciated so far, and let's see, is there also a pointer? Yeah. Is a gamma delta T cell, which is between 1 and 10% of all our cells in the blood. And these cells recognize, and this is becoming known in the last couple of 5 years, so-called butterfurulins. And it's not the classical way, what you heard this morning, you have a CD19 CAR-T, or you will later, which is recognizing a tumor. They're recognizing a conformational change in the cell membrane of a tumor cell, which is active in more than 50% of all cancer cells. So meaning with this principle, in principle, you could detect 50% of all cancer cells just with a single receptor, which seems to be very attractive and which also does not depend on mutational load. For those who are interested, the important thing is in these all the receptors that you break the balance, because our nature is made for cancer immune surveillance, and also our immune system adapts very quickly. That's what we heard later. I mean, tumor is constantly evading. It's a moving target. And the immune system, unfortunately, also is then adapting with the moving target and no longer recognize cancer. And by engineering sort of an imbalance in the system, you will always be able to target this cancer cell. This resulted in a concept for those who are interested, which is sort of like a CAR-T, where you have an alpha beta T cell, a CD4 and CD8, and you introduce a receptor, but not, as heard also later, you put in a CD19 targeting CAR-T, you put in this gamma delta T cell receptor because it has the potency really not only to go to liquid tumors, not only to hematology, but also really to go to most of the solid tumors. So for that reason, there was a lot of interest in doing so. We started a company a couple of years ago, but of course there was a big challenge. So when I came back from Seattle, starting in Utrecht, I say always missing the plane to Germany, as I'm originally German. We had a first publication about the new treatment modality. I had a department head coming from a company. He said, okay, first publication, you missed to write a patent, but you're doing a second publication, please write a patent. So that's the first thing we did. We write a patent on the new lead. We made a company. I was scientific co-founder of this spin-off company, which was recognized by Nature Biotechnology as one of the top 10 spin-outs at this time. Of course, you cannot buy anything for it, but it was a nice appreciation of what we did. We had a deal with Kite Gilead in 2018. There was another deal. That's how companies go. You sell it to the first company, then the next company is coming in. We had an Immunocore deal with Clade Therapeutics, a deal. And in the meantime, we were starting up the clinical trial. First in human trial, first publication, 2015, we said, this is something we need to put into GMP environment. It needed us 4 years for the preparation of the documents for submission and getting approval by the Dutch authorities, which were, by the way, very collaborative. And my take-home message from talking to regulators is, and I'm pretty sure you might confirm, they're not our foes. They want to understand what we are doing, and they need to be fed with information to make a well-informed decision on what they are doing. We had the first in human dosing in 2019. 224, it's a third dose level, and somebody who's here from a company would say, this is not at all a competitive timeline, and this is correct. So my take-home message of this one is totally exciting, because I brought my own idea I had as a postdoc back to the Netherlands. I started the company. I knew how it is to write patents at the end of the day, to negotiate with investigators to make it happen. But timelines are simply not competitive. So maybe I can do this one more time in my life, and then it would be done. So, and this is for companies not interesting. So what is the way to improve this? And then we had clinical outcomes. So was it the idea interesting, yes or no? And first dose level was okay-ish. Second dose level, we saw a complete remission, which is quite remarkable in this new concept. And then we were struggling with our own facility. It took a long, long time. And at the end of the day, we decided, because we had already the next generation developed, it's no longer competitive. You would like basically to go with the next concept with a new company again on the market, which is work in progress now. But we need to innovate and develop this process. And in the last year, I was very lucky also to work a lot together with Dutch government funding, and we initiated something that's called ONCOD Accelerator. And today I would like to also pique your interest in not only how important it is to make the right clinical trial, but how important it is to make the right decision which of your lead is reaching the clinical trial. Because I can tell you at the moment within my lab, I think 50 different leads, where we design it a little bit differently. But the key question is, on which one do you put your money to really bring it in a phase 1 clinical trial? Because we know also that pharmaceutical companies are not, like in the early days, just jumping on each idea. They nearly expect you to have a completed phase 1 trial, to have some activity data to see where it goes. And the strategy the Netherlands developed and really put 600 million euros into that, and we are lucky to be part of this, is to have different product pillars: small molecules, biologics, cell and gene therapy, where I'm co-leading it, and therapeutic vaccines. In addition to that, they said, okay, these are the different things patients could get. We need innovation platforms. The one is patient cohorts, where we have very well-defined patient cohorts with registers data, where we have also, for example, organoids next to it. That's the next pipeline, tumor organoids from healthy and diseased tissues, so that we can test on these tissues whether it's really recognizing. And of course, we need an AI next to it, because there are so many data out there. We need really to be able in a different way to master with this data. And just out today, I think if you look at Nature, there are 3 publications on AI-driven research. I think really that's a new way how to do this. Plus, don't forget, and this I think is forgotten by many basic researchers, so in this, and then we can make demonstrator projects on this. But the most important thing is you need to take along the regulation innovation along with it. Because just a very simple example, if you do with the retrovirus, which I still think, although less popular with regulators, you need to think about, do you make a master cell bank, or do you make another transduction, or do you go to lentivirus? Because if you make a wrong decision in the very beginning, the regulators will later say, yes, sorry, but this is really not what we want to see. So really engaging from the very beginning is one of the key things. And as regulation is really developing and people need to be informed and are also open for innovation, this is really very important. This is sort of led together with the NKI at our center, and then we have other centers in the Netherlands, and the Radbouw and the UMC Groningen, who's working together with this, where we really have substantial funding to bring things also forward. The major challenging in creating new CAR-Ts is that it's sort of, like I say, always like playing Lego. So I don't need any computer games. I don't need any other entertainment, because you can put it in different perspectives together. And then the question is, what's the best model to predict what is entering the first clinical trial? Because, of course, CD19 BCMA is relatively easy, although it depends on the antibody, but the targets are very well characterized. The targets we have are super novel. New targets are coming up, but are they really good targets? And the way how ONCOD Accelerator would like to sort of foster the interactions that you say you get funding, but you need to mix and match. It's like if you build your house or if you dress up with clothes, you go with the old one, but also something new one. And by combining, you create power. And we take in this case cell and gene therapy, we take organoids along, and then we take AI, or people would say bioinformatics, along and combine this to make well-educated choices. And just to show you one example, that's what we did together with Anna Rios from the PMC. And interestingly enough, she's working on breast cancer. And the yellow thing you see here is a conglomerate of 20, 30 breast cancer cells. And this little one here, that's a so-called TAC, what I showed you before. That's an engineered cell designed to kill breast cancer or colorectal cancer, but also AML and non-Hodgkin lymphoma. And then this one cell is coming, and only this one cell is docking to this 20, 30 tumor cells. And then you see these things coming, so it's spreading in tentacles, and it starts slowly biting into this process of tumor cells. So this takes hours. Yeah? And then you see slowly that these cells turn red, so they die. So basically, the power of such a cell is to really, it's David against Goliath, to really tackle an enormous amount of solid tumor cancer cells. And then at the very end, you see that such a cell is doing what everybody is doing after a lot of work. It's resting. But the big question for immune cell is, are you resting and dying? Or are you resting and then go back to your job? And a way how to further work on this was then the next paper we did, where we then looked at co-stimulation, because it's about in life also, how are you stimulated? So thanks for the invitation. I was already a lot stimulated by talking to different people, and then getting new ideas how you deal with EI or whatsoever. And that's the same for an immune cell. You need a sort of co-stimulation. We showed if you make the right co-stimulation, just looking at color coding, going back, you might see that the color coding from this to this is changing. And first of all, that in the initial population, a CAR T cell is not one type of cell. It has a lot of different properties. That's also what you expect. But once you get a co-stimulation, you see that this color coding changes. So each cell behaves a little bit different. And the biggest challenge is now to understand which of our 50 constructs is the one to put forward, where we would like to convince funding agencies to give us money for the first clinical trial. And we developed this concept. Conflict of interest are trying to set up now a company on this, because we need a lot of funding to really make industry-sponsored trials, because my belief is I have a lot of respect, and Julio Delgado knows, and other colleagues from Barcelona, a lot of respect for the efforts to make sort of existing leads as academic leads, because I think that's very important to create access. But I think our role in academia is also to develop new leads. But you can imagine going to solid tumor is a big business, so we won't ever be able as an academic institution to really roll it out. So we need a healthy interaction partnership with pharmaceutical companies. And you can see here sort of the first product which made complete remission in AML was in the model like this, and the new product is just creating cure. So we have a lot of hope in this one. But the question is how to bring this forward. And next one is, I think, which is very important also there to solve, and we heard a lot about this, is creating cell and gene therapy, well-defined patient cohort, and AI. And one way to do this, because I talked also here to some registry people, is to connect registries with also national registries. And I'm sort of part at the moment chairing the CTI working party of EBMT. And we really would like to have the Dutch CAR T registry, which is an isolated CAR T registry. Descartes from France is an isolated CAR T registry. How can you connect this data? And this is something we have now people working on, building modules to make this happen. So, but at the end of the day, as a doctor in the hospital, you type in once the thing, AI is scrapping then the information from the dossier from the patient. This is delivered to the Dutch registry, and the Dutch registry gives it to the EBMT registry. So like this, you really reduce the workload and increase the quality of the data in such a registry. Another thing is you need to define what data you would like to capture, which is not an easy thing, because I think every CAR T registry that developed over the time defined their own ideas what it should be. And we started a harmonization project, and you see also T2 Wolf is on that. I had a discussion on T2 Evolve where we discussed about harmonization of this data. What are the data in there, which I think is for all the clinical trials is important. And we now are in the process of publishing this, where we have must-haves and nice-to-have, and where we would like to inspire, on the one hand, existing registries to see what choices they made. Do these choices fit with what the public thinks? For example, I know now already that the EBMT is collecting a lot of data nobody is interested in. Should we just stop so? We are also collecting some data everybody likes, that's perfect, but also some data we are not collecting because we thought it's not important, but apparently everybody finds it important. And like this, every registry makes own choices, and now the presentation stops, at least here on my screen. So that's the one thing. And the last thing I would like to make, 30 minutes, that's a short time, but I will try to make it, is the production part. Because one of the major challenges, these are clinical trials for CAR-T in Europe. We made a survey, all centers in there, and this is the density of ATMP facilities per country. And you see that there's an enormous difference between different countries, which also reflects back to the number of clinical trials you can do. And one of the major challenges in the future will be, are we able to align together our resources? Just to give you some key data on that, we looked at how are these facilities in the facilities, how is the staffing in there? You need a lot of people to do this. And something I try to advocate for now is, can't we share our resources? For example, a pharmacist can work in Barcelona, but help also the facility in Utrecht. And I just had yesterday a nice talk with one of your old colleagues from France, who's working actually in Prague, but managing whole Europe. And why shouldn't we be able as academia to do so, to share these resources? But of course, the quality systems are different. This is something I think we should really develop together, because it's not only about the facilities, which are the limitation. The true limitation for each country is knowledge and is skills in people. And this is a survey from a pharmaceutical company where you clearly see that certain skills are just not available. And if it's for a pharmaceutical company, where we have big pharmaceutical companies in Utrecht or in the Netherlands, difficult to find these people. As academia, it's even more impossible. So we need to share these people. And you see also that these people, that's how long does it need to hire them? And then how long do they stay there? They come and they go also again. So we need to find a model together as academia. If you would like to run clinical trials, hospital exemption, how do we share these personnels? And just because of time, and even think about, if Barcelona is doing great production, maybe for one product, but UMC Groningen is another great production, maybe for another product. We need to think through where we would like to go with these approvals. Is it only for our country? Is it for 2 countries? Or is it really for whole Europe? I think it should be for whole Europe. And I think also we should not necessarily compete with all the pharma, but we should find a healthy ecosystem where we find our own role. Pediatrics is super important. How to do this for pediatrics, and what should be done for also not by us. A lot of things should not be done by us, to be honest. Conclusion, and then I nearly made it, 1 minute 50 seconds too long. So academic translation requires absolute professionalization, also in the business cases. So when we calculate the amount of money and we have funding problems for the net clinical trial, we should just 30%, or maybe even 50% add to the product price, because we need it for the next product, because companies are also continuously going on with the next product. We need to integrate regulatory considerations early. I mentioned the backbone we are using. Manufacturing capacity remains really uneven across Europe, and need to find a way to connect this. And the main bottlenecks are, in my perception at the moment, not facilities, but people. People with the right knowledge to get the job done. And this means we really need to coordinate this. And luckily, I mean, there is some hope. So this has been mentioned a little bit. We need expand existing regulatory support. This we have harmonized regulatory frameworks. The sandbox was meant, central authorization we'll hear about. And the proposed European Biotech Act maybe will make it happen. And now I finish and thank everybody.
Thank you.